effector organs of insects. Primarily, the sound producing organs and light producing organs. So, first, we are going to learn about the sound producing organs. Insects of some species are capable of producing and perceiving sound with the help of variable but well defined mechanisms and organs. Their sound has a certain biological significance that it may be used as a species specific language amongst the opposite sexes and individuals as a whole during mating, nutrition, danger, etc. It is also found that an individual insect can produce different sounds of variable frequencies in accordance with their requirements. In some insects, the sound is incidentally produced as a byproduct of some routine activities, as wing beating in case of Drosophila apis mosquitoes, wing hitting the body, Schistocerca and other grasshoppers, and the sound of humming or buzzing type of 250 to 5,000 cps in various species of insects. Moreover, the sound is produced by merely tapping or hammering the substratum. Then death watch beetle, Anobium bark beetle, tenebrinoid beetle, Polthrus, and termite soldier of Zootermopsis, Reticulitermus, and Leucotermus species. There are some Sucoptera and Plecoptera in which sound is produced by tapping a terminal abdominal knob against the ground. Special mechanisms of sound production. In well-known sound-producing insects, the sound is produced by well-developed sound-producing organs, though they may differ in sound-producing mechanisms. The main sound-producing mechanisms are of three types: stridulatory mechanisms, timbal mechanism, and epipharyngeal mechanism. Stridulatory, timbal, epipharyngeal mechanisms. Stridulatory or frictional mechanism. A large number of insects produce sound by the friction of two parts or organs of the body against each other, and such organs are known as the stridulatory organs. Stridulatory, friction based. Okay, they are well developed in some species of Orthoptera, Coleoptera, Dictyoptera, Hemiptera, Lepidoptera, and Hymenoptera or ants. The stridulatory organs may differ in location from one species to other, but basically they consist of a file and a scraper. These organs are confined to only male insects of the orthopteroid species or families like Tetigonidae and Tigrillidae, and so that only males can produce the sound by efficient stridulation. Stridulatory organs. Both the stridulatory organs are formed due to secondary modification of the cuticle. The file is formed of a row of teeth, ridges, or projections. The scraper is composed of hard knobs of pegs arranged in a linear fashion. The file and scraper are also termed as strigil and plectrum. Strigil and plectrum, respectively. In both the sexes of cockroaches, the stridulatory organs are confined to pronotum and on coastal vein of the tegmen. In the Orthoptera, there are two sets of stridulatory organs in grillets, while only a single set in Tetigonids. On the elytral vein, the file is present on the first anal vein, while the scraper on the margin of the tegmen. In Acaridids, moreover, the file occurs on the outer surface of each tegmen and scraper on the inner side of each hind femur. In Hemiptera, the stridulatory apparatus varies in location. It is abdominotibial, Toxoptera being the thoracic, being abdominal or abdominofemur stridulatory apparatus. In Coleoptera, stridulatory apparatus is retained by larvae, in which spines on the maxillae are rubbed against the striated plate on the mandibles. Stridulatory mechanism of sound production has also been observed in the pupae of Orectus due to friction between pupil and adult cuticle. Adult beetles of several species, mostly males, have stridulatory organs on the abdomen, femur, head pronotum, thorax, abdomen, elytra, etc. File on one region while scraper on the other. The Thycophora, the file is situated on the hind wing and scraper on the inner side of the hind leg of males. Opener muscles, forewing, wing movement, closer muscles. In Hymenoptera, among Ponerine, Dorline, and Myrmecinans, dorsal side of petiole consists of a ridge which acts as a scraper, and it rubs against a series of transverse striations lying at the base of the first tergite of the gaster. Mechanism. It involves friction of scraper over the file, and particularly in Orthoptera, the mechanism is elytral type in male gryllus and pectigonax, while elytrophemoral type in acridids. The elytral method. One elytron overlaps the other, and the insect moves the elytra laterally across one another, forcing a scraper to stridulate over a file. The movement of scraper over the file causes wing membrane to vibrate, to which the file is attached, so that the chirping sound is produced. The frequency of sound produced by this method in grillets is very low, about 2 to 10 kcs, while in Tettigonids about 5 to 100 kcs. During sound production, the elytra is raised to an angle of 15 degrees Celsius to, 50 degree to 40 degree to the body, and then they open and close so that the scraper can rub on the file, causing an electron to vibrate and to produce a sound. The sound is produced on closing the elytra and not with opening. Each closure of wing produces a single pulse of sound. An elytrophemoral method. Irrespective of sex, the insect moves hind femur up and down, and the pegs rub over the elytral file, which causes the elytra to vibrate and the sound is produced. The frequency of sound is high. The next, so this is all about the stridulatory or frictional mechanism, where strigil and splectrum are involved. Next, the timbal mechanism. The timbal organs are well developed in some Hemiptera and Lepidoptera. The timbal organs are highly evolved in Hemiptera, Archaeorhyncha and Cicadas, Pentathomids among Hemiptera, and Arcticidae and moths in Lepidoptera. Then different parts. Timbal of cicada, cross section of the first abdominal segment, and we try to learn about the parts. You can see a portion called a tergum, rim of timbal, air sacs, hemocoel, viscera, timbal cover, timbal, apodeme, timbal muscles, rim of timbal, then tensor muscles, air sac, folded membrane opercula, compression, stert and sternum hemocoel. The timbal organ. In Hemiptera cicadas, the timbal organs are well developed in the males as a pair of membranous structures on the dorsolateral regions of the first abdominal segment. The timbal is a chitinous membrane. It is supported around its edge by a strong chitinous ring, and it is normally bowed outside. The timbal muscle is attached to the timbal at the center. Internally, it is communicated with the air sac, which opens through a spiracle. In Lepidoptera, the organs are paired structures situated on the lateral side of the metathorax. The modified catepsternum of the metathorax is swollen outward and sclerotized. There are striations about 10 to 60 in number towards the anterior end of the timbal. The metathoracic basilar muscle is an actuating muscle. In Heliothisia, presence of a large number of microtimbals are reported. The timbal organs of these moths are also known to produce ultrasonic signals to confuse the bats. When the timbal muscle is contracted, the timbal is pulled inward, and a subsequent distortion of the timbal membrane produces pulse sound. With the relaxation of timbal muscles, the timbal springs back to its original position due to its elasticity, and it again produces another pulse of sound. Two timbals work synchronously. Timbal of Melisine mesothoracic segment. Basilar abdomen, then anasternum, anepisternum, timbal, striped band, catepisternum, part of timbal covered in scales, meron, epimeron, coxhalf leg. Epipharyngeal mechanism. Third one, epipharyngeal mechanism. Epipharyngeal mechanism, it is found in the death's head hawk mouth. Epipharyngeal apparatus. The proboscis acts as an amplifier. The pharynx as an air pump. Epipharyngeal mechanism. Epipharyngeal apparatus. The proboscis acts as an amplifier. The pharynx as an air pump, and epipharynx as a vibrating instrument. The pharyngeal muscle operates the air pump pharynx mechanism. Contraction of the pharyngeal muscles induces dilation of the pharynx and air is sucked through the proboscis. It causes the epipharynx to vibrate and a sound is produced by a pulse of airstream. Pharyngeal muscles, pharynx, epiphanary, esophagus, salivary gland, proboscis. Pharyngeal cavity, pulsed airstream, epipharyngeal vibrating, base of proboscis. Expiration, pharyngeal cavity, epipharynx held erect, then exhalant airstream. Relation of the pharyngeal muscles induces contraction of the pharynx and air is blown out. The exhalant airstream also produces a high-pitched sound like a whistle, during which the epipharynx is held erect. Sound production is possible only if the pharynx is empty. Characteristics of insect sound. Man can hear sound of some orthopteran species that of acridids from a distance of 1 or 2 meters, Nemobius from 8 meters, Tetigonia from 100 meters, and that of Gyrolothalpida from 350 meters. First vibratory apparatus to a single to and the flow movement may produce a single or a series of pulses. The pulses show a unique sequence called a phrase. The sound may therefore be a continuous or discontinuous with silent intervals. The pulse can be defined as a discrete or discontinuous tail of sound waves. Frequency. The frequency of sound is specific, constant in a particular species. The frequency of sound produced by some species of insects. Honeybee, 250 C per S. Classification and significance of sounds. Besides the definite frequency, the sound of a species varies in its characteristic, mostly due to the pulse repetition frequency. The oscillograms of the sounds produced by different conditions by some orthopteroid insects show that each species produces a number of different songs having different biological significance. On the basis of oscillogram and functional specificity, the insect songs are classified by Alexander into various types such as calling, aggressive, courtship, warning, copulatory, post-copulatory, and recognition songs, etc. Mechanisms of sound production: calling songs, aggregation song, courtship song, second phase. The mechanism of sound production and reception is well established in Orthoptera, Hemiptera, Coleoptera, and Diptera. The sounds are used in promoting mating, courtship, and copulation among opposite sexes, exhibiting unique acoustic signaling as an integral part of reproductive behavior. The acoustic signals in reproductive behavior are initiated with the calling song in order to bring the opposite sexes together, and then followed by the courtship, copulatory, and post-copulatory signals in a well-planned fashion. The acoustic signals are species-specific and perhaps integrated with the extrinsic as well as intrinsic factors influencing either directly or indirectly reproductive physiology and behavior of the insects. Acoustic behavior is also found in rivalry and territoriality, adapted Orthoptera, and in social termites, ants, and bees. It is involved in reproductive, defensive, and in various purposes, forming a sort of an operative acoustic communication system in the social insects. In the arcid and Cheniosid modes, the sound is used as an acoustic mimicry like bats. So that's all about sound producing organs. Then next, light producing organs. Bioluminescence is reported from a large number of insects. True or self-bioluminescence occurs in only some species of insects belonging to the following orders: Columbola, like lighting springtails, Hemiptera, Homoptera, lanternfly, then Diptera, larva, glowworms, Then Coleoptera, fireflies. Okay. The luminescence, so different types. Lighting springtails, lanternfly, glowworms, fireflies. The luminescence may occur in both sexes of it, may be restricted to female and even up to the immature forms, particularly in Lambridae. In insects, the light is produced by the special light-producing organs. The light-producing organs are situated on the ventral side of the body and are developed from the fat body. They, however, differ in their number and location. In Columbola onychurus, the light is produced from whole body and the hypodermal fat layer functions as a photogenic organ. In Coleoptera, mostly Lambridae, the organs show wide diversity among the male, female and larvae. The light-producing organs are situated on the ventral side of the body and are developed from the fat body. They, however, differ in their number and location. In Columbola onychurus, the light is produced from whole body and the hypodermal fat layer functions as the photogenic organ. In Coleoptera, mostly Lambridae, the organs show wide diversity among the male, female and larvae. In the male of Photurus, there are two pairs of photogenic organs situated ventrally on the 6th and 7th abdominal segments, while the female possesses only a single pair of organ in the 7th segment. The larvae, moreover, bear no doubt a single pair of organs, but they are located on the 8th segment. Among Diptera, the luminous organs, Blitophylla, are the exceptionally enlarged tips of all four malpighian tubules lying in terminal segment of abdomen, while in others they are derived from the ventral hypodermal fat layer. Platura bears a pair of photogenic organs at the caudal end, while the whole body of larvae and pupae of Ceroplatus is luminous. A large number of organs is reported in some sort of Coleoptera. There are three organs, a pair of lateral side of pronotum and a large median and middle of the first abdominal segment in Electridae. The larva possesses three photogenic organs, two lateral and one median, one on each abdominal segment, while the star bones bear a single at the center of each abdominal segment. In Phyrexothrix, railroad worms, lateral marginal photogenic organs are located on the 2nd thoracic to 9th abdominal segments, which are emitting the blue light. Besides these 9 pairs, two additional photogenic organs emitting red light are situated on the head. Studied in detail the structure of light organs of Photurus. The light organs consist of the photocytes, large sized cells lying just beneath the epidermis. They form cylinders running at high angle to the cuticle, measuring about 10 mm thick. The dorsal cells form a layer over the photocytes, and cuticle overlying the light organ is transparent. The photocytes are richly supplied with tracheal nerves. Each trachea gives off branches at right angles, which break up into a number of tracheoles running between photocytes parallel to the cuticle. The trachea are placed at 10 to 15 mm distance from each other, providing a short path for O2 diffusion. The tracheal origin is enclosed within a large tracheal end cell. The inner membrane of the tracheal end cell binds the tracheoblast and is completely folded. The nerves entering the photocyte cylinder end as the spatulate terminal process between the plasma membrane of the end cell and the tracheoblast within which the tracheole arose. The terminal processes possess two types of vesicles: large vesicles of 1000 angstrom pneumosecretory droplets and small vesicles of 200 to 400 angstrom. Trachea, nerve, lumen of cylinder, end cell, tracheolar cell, tracheole, dorsal layer cells, granules, waste granules, photocyte granules, photocyte, mitochondria, epidermis, transparent cuticle. Okay. Large vesicles and neurosecretory droplets and small vesicles or acetylcholine diameter. The photocytes are packed with photocyte granules. The granules contain a cavity connected with the outside cytoplasm via a neck. These granules contain photogenic substrate, luciferin. Small granules occur dorsally and ventrally. Mitochondria are sparsely distributed in the photocytes. The dorsal layer cells contain urate granules and the cells form a reflecting layer. They also store the oxyluciferin irreversibly produced in light production. In photorests, two photogenic organs together contain about 15,000 photocytes, forming some 6,000 cylinders, each with 80 to 100 end cells. 80 to 100 end cells. Mechanism of luminescence. Light is produced by the oxidation of luciferin in the presence of the enzyme luciferase. Luciferin is first activated by ATP in the presence of magnesium and luciferase to produce acetyl luciferin. The acetyl luciferin is oxidized by an organic peroxide, again in the presence of luciferase, to form so-called excited adenyloxyluciferin, which decays spontaneously to low energy adenyloxyluciferin with the production of light. So, light is produced by the oxidation of luciferin. 2C4-carboxythiazole 6-hydroxybenzothiazole cysteine in the presence of the enzyme luciferase. Luciferin is first activated by ATP in the presence of magnesium and luciferase to produce adenylyl luciferin. The adenylyl luciferin is oxidized by an organic peroxide, again in the presence of luciferase, to form so-called excited adenyloxyluciferin, which decays spontaneously to low energy adenyloxyluciferin with the production of light. The energy of this process is obtained directly from the oxidation process, not from the ATP, and it is released in one large step. The radiation is very efficient, about 98% of the energy involved being released as light. The low energy adenyloxyluciferin produced inhibits further reaction by binding with luciferase. The pyrophosphate, however, removes the inhibition. When the light organ is stimulated by a nerve, the acetylcholine released at nerve ending react with ATP and coenzyme to yield pyrophosphate. The pyrophosphate diffuses to the photocyte granules and stimulates the production of light by removing the inhibition of luciferase. The chemoluminescence reaction is given below. Light. 1 mole of oxygen is consumed per luciferin molecule, producing 1 quantum of light. According to Lee and Mallory, the pH induces change in the luciferase structure in such a way as to give 2 colors of light. The color of light produced by various insects is given below: yellow, green, lamberis, blue-green, Bolithophila, white, fulgora, green, red, Phylloxothrix. The variation of color of light emitted by different species of fireflies is due to luciferase and not that of luciferin. Proton obstruction is essential for obtaining the yellow-green light. 2 different ionization states of luciferase and the subsequent effect on the bound emitted would appear to be responsible for the emission of 2 different light colors, red and yellow-green. Low pH causes decrease in intensity of yellow-green light, leaving a dull brick orange glow. Such as an alkaline and neutral pH 7.6, single emission band in yellow-green region, and intermediate pH red emission band appears at 6.6 nm, and at pH below 5.5 red emission band. Yellow-green completely suppressed. Change in pH takes interaction of the enzyme with the benzothiazole OH group or the amino group of AMP, altering the effect of fluorescence of chemoluminescence properties of the bound intermediate, which is essential for light emission. Variation in temperature, ions Mg++, Zn++, Cd++ or Hg++ and urea capable of shifting yellow-green to red color. Mode of light emission. There are various modes of light emission giving different glow patterns as described below. Like continuous glow. Light is produced without interruption in a continuous glow of uniform intensity like clavicorn female of Figogodes. Then intermittent glow. The glow lasts only for a few seconds. Light appears and disappears in a second. Then pulsation glow. Light is emitted in pulses. Flash of glow. The flash consists of a burst of light which remains only for a short duration with greater intensity. The light intensity in a flash rises abruptly from 0 to maximum and declines suddenly back to 0. Duration of flash is of the order of 0.1 to 0.2 seconds with only one peak of maximum intensity and assisted by multiple ranging peaks. The flashing pattern is specific to species. True fireflies such as Fortinus, Photorus, Lucicola, etc. Control of bioluminescence. Two principal theories of neutral control have been proposed. Structural neurocontrol theory. It is advanced by Snell and supported by Alexander. It holds that the nerves regulate the opening of a valve in the tracheal end cell, which controls the inflow of oxygen, a key factor known to involve in light production. But criticized the theory on the basis of well-known fact that light-producing cells are virtually anaerobic during the long non-flashing periods, and non-mechanisms of oxygen diffusion in insects could not support the kinetics of flash response. Buck in 1955 considered the neutral influence might act at the enzyme level, and all the reactants necessary for luminescence are released to react by neural activity. The luciferase neutral control theory. It is proposed by the neutral activity causes an indirect release of luciferase which react with the necessary substrates, all of which are present in excess. The flash is terminated when the enzyme is again sequestrated in an inactive complex. The light organ is a true neuroeffector organ. The brain initiates the flash. Brain and peripheral neuron complexes have inhibitory effect, while abdominothoracic ventral nerve cord has excitation effect. The inhibitory substance may be adrenaline in adult, a neurotransmitter, a monophenol resembling phenolphine in larvae. Significance of light production. In lamprids, the light is a mating device to attract the opposite sex. In the fireflies, where both males and females have luminous, a fairly complicated signal system has been developed. Each species of firefly has a characteristic flash to which the female species can recognize. Fortinus pyralis males fly above 50 centimeters from the ground and emit a single short flash at regular intervals. The female climbs above the grass and flashes only in response to a flash of light which is produced by the male. The female responds to male producing light within an area of 3 to 4 meter radius. The male then runs directly towards the female and glows again. The female and male then emit repeatedly 5 to 10 times until the male reaches the female and mates with her. In some fireflies, only one sex female is luminous, broadcasting a species-specific signal to the non-luminous male. Flash pattern of various species of Fortinus ardens. Fortinus obscurus differs from variable intensities and responses from the female. In Electridae, females emit bioluminescence and pheromones so that males can be attracted. The luminescence or Blittophila larvae serve as a lure, attracting insects on which they feed into networks of glutinous silk threads which they spin initially. Bioluminescence research is also being conducted for use in the medical field. Virologist Christopher Contag and Pamela Contag have begun using bioluminescent bacteria to follow the progression of infection in mice. Researchers inserted bioluminescent genes into Salmonella bacteria, causing them to glow. They could watch as the infection spread and could judge which inhibitors are most effective by observing the reduction of bacteria. Although this false basic virus makes gene insertion more difficult, studies have been initiated to attempt to track the progression of the AIDS virus by changing the cells of the animal to glow when the virus invades. It is hoped that bioluminescence will be used to track the location of cells altered with gene therapy. This research could also illustrate whether these cells were producing the proper protein after modification. Woodland husky of Harvard University anticipates technological advances allowing surgeons to locate the cancer and know where to cut more effectively than their present techniques. By using firefly luciferase, biologists can ascertain the amount of adenine triphosphate ATP in plant, animal, and bacterial cells. ATP acts as a stored energy for those cells and is directly related to the quantity of cell present. In one application of ATP indicated the indices that amounts of bacteria present in blood or urine samples. Jellyfish acronym uses calcium instead of ATP for bioluminescence, so this acronym is used in a similar way to determine the amount of calcium present. Bioluminescent organisms can determine toxicity because the noxious substances reduce the glow by killing the bacteria. Current research has found success in identifying eye cancer and breast cancer with this technique. In one case, a bioluminescent bacterium called Vibrio fischeri developed sheds light on how to combat infectious diseases with something called quantum sensing. This is essentially bacteria's ability to sense and communicate with other bacteria. For the bioluminescent software triggers a rather astonishing light display, but a similar kind of communication can be observed in disease-causing bacteria too. Bioluminescent bacteria may help in detecting food poisoning also. It is mostly caused by cholera bacteria. Vibrio cholerae, they are gram-negative road-like organisms. They are facultatively anaerobic, which means they can survive either with or without oxygen. Using bioluminescent techniques made this fascinating investigation possible. Thank you.